In our final experiment, we're going to be using plasmids to express the protein GST SH2 in E. coli cells. So we're putting together everything that we've learned in the semester. We've learned about plasmids. We've learned about how to do a protein purification. We've learned about growing E. coli cells. We've learned about centrifugation. And we're going to be applying many of these techniques to express our GST SH2 protein. Some background on protein expression. There are many barriers to obtaining fully active, properly folded proteins. One of the barriers is when you're trying to make a whole bunch of protein all at the same time, sometimes you're making it so fast that there's not enough time for the proteins to properly fold. So if this is a properly folded protein and you're making it too fast, sometimes the proteins get misfolded. When you have a large amount of misfolded proteins, these are going to make something called inclusion bodies. So these misfolded proteins where the charged moieties, the charged amino acids, may not be properly to the outside of the protein. Maybe the hydrophobic amino acids, for example, are to the outside in a misfolded protein. They will aggregate together in these inclusion bodies to try to bury the hydrophobic residues away from the aqueous solution. And the amino acid sequence might be correct in these proteins, but they're lacking something. They're lacking the proper folding ability to make them fully active proteins. One of the things that can cause inclusion body formation is a lack of chaperone proteins. Sometimes when proteins are being made, they're starting from one end of the protein, they're being expressed. If they're being expressed too fast, such that part of it does not fold before the whole thing folds, this can cause an inclusion body. If they don't have signals to have proper post-translational modification, maybe they need a secretion signal because it's going to be it needs to go through the membrane, or maybe it needs a kinase to be able to phosphorylate it for proper activity. So if there are not enough of these auxiliary proteins, you will again not be able to have a fully active protein. Sometimes your barrier is just that you have poor expression levels of your protein. You might not have a good ribosome binding site so that you're not getting good transcription and therefore not good translation to make a lot of your protein. Or perhaps your protein is lethal at a high concentration. One of the things you need to worry about if you are trying to express a lethal protein is, how am I going to be able to grow my cells to a high enough concentration if my lethal protein is stopping my cells from growing to that high enough concentration? And we're going to be talking about ways to do this. Or maybe you only have the problem of being able to purify your protein after it's all been made. So there are some solutions to some of these problems. So if your problem is that you don't have the auxiliary proteins, or lack of auxiliary proteins are causing inclusion bodies or just inactivity of your protein, one thing you can do is you can identify and co-express auxiliary proteins. So for example, if you're lacking a kinase, at the same time you were trying to express your protein, you could also be trying to express the kinase, and then the kinase could go and put a phosphate in the proper position on your protein. But do you know what the problem is with your protein? Why it doesn't have activity? That's the first barrier you'd have to overcome. And then the second barrier is, do we have the proper gene for the kinase? And is that kinase going to function properly? That can be a PhD dissertation all in itself of just trying to solve this huge problem. One thing that can be done if you're maybe lacking some proteins is you can try to express your protein of interest in yeast or eukaryotic tissue culture cells. So sometimes you have a protein that will not express well in bacterial cells. When I was working in the lab, we had this problem. To make a active complex of the 3 proteins we were trying to express that needed to come together to make that active DNA binding agglomeration, we needed to have 3 separate proteins that were expressed as 3 different genes. And we tried expressing them all in the same bacterial cell, but one of them was a lethal protein and did not express very well. And we tried expressing them separately. We tried expressing them together. We tried a lot of different ways. And we just could not get all 3 proteins to express to high levels at the same time in the bacterial cells. So what people do sometimes is they would try yeast. They don't want to go directly to eukaryotic tissue culture cells because eukaryotic tissue culture cells are very, very expensive to maintain and grow. It can be hundreds of dollars a week just to keep your cells growing, and not everyone has that kind of money. Bacteria, on the other hand, are cheap. If you're able to express your protein in bacterial cells, you can, put your bacterial cells in a glycerol stock, mixing growing cells with glycerol, and stick it in the freezer for a couple of years, and then pull it out when you need to make more. Versus, tissue culture cells, you have to keep it in liquid nitrogen, and then you're trying to maintain viability, and so sometimes people will use yeast as an intermediate step in there. But if bacteria does not work and yeast does not work, then very often growing your protein in eukaryotic tissue culture cells is the answer. That was certainly the answer for the sets of 3 proteins that we were trying to make once we expressed them in eukaryotic tissue culture cells rather than bacterial cells. All 3 proteins were able to come together as a complex. Sometimes people will make a fusion protein with a cleavage site. So if your problem is that you're not getting a lot of properly folded protein in solution, you can start by having a very highly expressed protein that will drag your protein with it when it goes into solution. And if you have a cleavage site, so that after your protein's in solution, you can cleave the highly expressed protein away from yours and be able to purify just your protein, that's another way of getting your protein in solution. So one of the ways that people do this is using the lac promoter. And I have a picture of the lac promoter in just a moment, but basically, when you have a lac promoter in your bacterial cells on your plasmid, you can add IPTG to it, and IPTG is a lac analog, and this lac analog then binds to a repressor and stops the repressor from binding to the operator and allows expression. So let me show you how that works, and we'll be back to the slide in a minute. So here is the gene that's making the repressor. So normally this gene is on, making the repressor, and the repressor is binding to the DNA, and it's stopping RNA polymerase from expressing the genes in the lac operon. Now, it is possible to remove these genes from the lac operon and substitute in your protein of interest gene right here, so that by cloning your gene in right here, this system will work to express your gene of interest rather than the genes in the lac operon. So Under normal circumstances, the repressor is binding to the operator and not allowing polymerase to function. Once you add a lac analog, in this case IPTG or lactose, it binds to the repressor and stops the repressor from binding to the operator. And when the repressor is no longer binding to the operator, then this allows the mRNA to be made, and if you have your gene in here, this is allowing the mRNA for your gene to go forward, make the mRNA, which is then able to express your protein of interest. So this is a very powerful system that many, many people use. So, as I said, this fusion protein drags your protein into solution, and this can be used to also purify your protein. So if the first protein that is expressed in this system is something like a maltose binding protein, and the maltose binding protein is hooked to your protein of interest, then you can use a column with maltose on it, and because it's a maltose binding protein, it's going to bind to the maltose, and then you can elute it much in the same way we did in the aldolase experiment, where we bound our aldolase to a phosphocellulose, because that's what fit in the active site, and then we added the fructose 1,6-bisphosphate to compete it off the column. Same thing can happen here. You would then add maltose to your elution buffer, and that would make the maltose binding protein that's hooked to your protein let go of the column. But meanwhile, before you add the maltose, you've washed that column, you've washed all the other proteins which are binding to the column off, and so you're able to purify your protein. Now, this also goes with glutathione S-transferase, and that's what we happen to be using. Our highly expressed protein in this case is glutathione S-transferase, and glutathione S-transferase has been hooked to the protein that we want to express, the SH2 protein. And so the glutathione S-transferase is very soluble, and it drags the SH2 into solution. Once it's in solution, we want to be able to purify it, and so we're going to bind it to a glutathione agarose column. Agarose is the base, glutathione's on it, and the glutathione S-transferase protein binds to the glutathione. When we have finished washing off all the other proteins that are not binding, like our glutathione S-transferase SH2 is binding, we simply add glutathione to the buffer, and that will compete off our glutathione S-transferase, and the GST SH2 will come off the column. We also have a cleavage site on there, and that cleavage site allows the GST to be separated from the SH2 after purification. There are even other ways of doing it. Calmodulin binding peptide is another thing which is going to bind to calmodulin fixed to column material, or you can have protein A, and protein A is going to bind to an IgG column. And these are not the only solutions. These are just some examples of what people have used. So, as I said, we are using GST, and GST is a 27 kD cytoplasmic protein that binds glutathione, and it will bind to glutathione agarose, so we're using that affinity chromatography that we learned about in the aldolase experiment to be able to purify our protein. If we wanted to, after our GST SH2 has been purified, we could use thrombin to cleave our GST from our SH2. So this comes in the form of a plasmid that's called PGEX. PGEX has an ampicillin resistance gene on it, so you would want to make sure that you are growing your bacterial cells containing PGEX in the presence of ampicillin, so that you can make sure that you are maintaining your PGEX in the bacterial cells. And it contains the LAC-I gene, which is making that repressor, which is coming over here and binding to the LAC operator. And in the absence of IPTG, you have lots of repressor binding the operator, stopping any protein from being made. As soon as you add the IPTG, it stops the repressor from binding the operator and allows production of the protein to go through. So in this plasmid, it comes with the gene for GST already in there next to the cleavage site for thrombin. And that's next to a multiple cloning site. We've talked about multiple cloning sites before. They just have all these different restriction enzyme sites, so that whatever restriction enzyme sites you might need to be able to clone your particular protein in there, chances are you'll be able to find a multiple cloning site, restriction enzymes, to be able to just pop your gene of interest right in there. So it's making the GST, it's making, in this case, SH2. When we want to separate it, we can use thrombin. We don't have to, and we're not going to in this experiment, but if you were really doing this in a lab, then you probably would use thrombin. And then there's a stop codon, so it just doesn't keep getting made forever. If we wanted to cleave GST away from the SH2, and we had already purified the protein, then to be able to separate the GST from the SH2, you can simply put it back over the glutathione agarose column, and the GST will stick to the glutathione agarose, fresh of course, not with free glutathione in solution anymore, and the SH2 would now go on through the column, because now when it's separated from the GST, there's no longer anything to make it bind to that column. So it is possible to express GST SH2, use thrombin to cleave it apart, and separate out just the SH2 protein and purify it. Sometimes people don't need an entire huge protein. If they just have a problem with wanting a quick way of being able to purify a protein, there's something called a fusion tag. And a fusion tag is a small stretch of amino acids that can be added either to the amino terminal or the carboxy terminal of a protein. Now, they don't generally increase expression levels or the solubility of your protein. So you could not use a fusion tag if you were having problems with inclusion bodies, for example. But if you just wanted to use it for purification, people use those a lot. And what it does is it encodes an epitope. So an epitope is just the shape that the antibody detects. So the stretch of amino acids, the fusion tag, encodes an epitope, a particular shape, which can then be detected by an antibody. So just in the same way we did with the GST, we would want to bind it to a column. So one example of a fusion tag is called 6-X-His. It's a polyhistidine tag, and that's going to cause it to bind to a metal chelate resin, and it can then be eluted off by competing it off the column. Another example is a flag peptide, which binds an anti-flag antibody resin. And as you might imagine, to get your protein of interest that contains the flag peptide off of the anti-flag antibody resin, you would just add more of the flag peptide to compete it off the resin. A Mc is going to bind C-Mc antibody resin. V5 epitope binds an anti-V5 antibody. You can have these in conjunction with each other. So when I was in lab, we used a set of fusion tags called flag, Mc, His. And we had a flag peptide next to a Mc peptide next to our 6 histidines. And we could use these for either binding general antibodies to prove that we had isolated our protein, and or purification. We could even do purification with 2 different types of columns to make absolutely sure that we were only getting our protein of interest when we purified it. Another set that we used is we would use a flag V5 hist, for example. So you can combine these in different ways. Another example is the S tag, which binds the S protein from RNase A. The HA tag, which is a hemagglutinin glycoprotein, which binds the HA antibody. So all of these things are just little short stretches of amino acids. They're around 10 to 15 amino acids, and you don't even have to remove them if they're not affecting the activity of your protein. And again, they can be added to either the amino terminal or the carboxy terminal. So if, for example, when your protein is being made, if the amino terminal happens to be buried in the center of the protein and maybe is not accessible to being used to bind it to a resin, you say, oops, well, let me try the carboxy terminal and see if that works better. So you can, you might have to do it a couple different ways to make sure. Now, in expressing our GST SH2, there were a lot of problems that had to be worked out by the person that worked out the protocol. And this was someone's master's project that had to do all this. They first transformed the plasmid into BL21DE3 cells, which is a strain of E. coli that's very good for expressing proteins. And then they had to work out how long to grow the cells and what temperature to grow the cells at. Sometimes if you're trying to grow cells at 37 and express your protein at 37, you don't get the best yield of protein. So questions that have to be answered are, should I grow it at 37? Should I grow it at 15 degrees? Should I grow it at 25 degrees? How long should I grow the cells? What absorbance should I grow the cells to before I add the IPTG? And when you add the IPTG, what concentration of IPTG should you add? 0.5 millimolar, 1 millimolar, 2 millimolar. So different amounts can express proteins differently. How long after you add the IPTG do you let it sit there before you spin down your cells and freeze them, ready to break them open to extract the protein? So all of these things can change with different proteins that are being expressed. So in other words, a grad student sat there and said, okay, well, I'm going to try this with 0.5 millimolar, 1 millimolar, 1.5 millimolar, 2.5 millimolar. And I'm going to say, what's the best amount of IPTG to add? And then they had to say, okay, well, after I add the IPTG, at what time do I get the greatest amount of protein being expressed? And you might say, oh, longer is better, but no. So what happens is there's usually a time course of if you take samples and run all your samples on a gel that they've been induced for 1 hour, 2 hours, 3 hours, 4 hours, 5 hours, 6 hours, you will see the protein increase, and then you will see the expressed protein start to decrease. So you need to figure it out. There is no one magical answer. After all that is done, and this is done outside of lab time actually by the teaching assistant who knows, what temperature to grow the cells at. We start at 37, and then after we add the IPTG, our teaching assistant actually has to lower the temperature to 25 degrees for a certain amount of time, add the IPTG, and induce for 4 hours. So they have to know all this. Luckily, it was already worked out in advance by the people that gave us this clone. So after this has been done, the cells are spun down and frozen, and when you see the cells in class, they are resuspended in lysis buffer. And remember that lysis buffer basically is a tris buffer and some EDTA to chelate those divalent cations, some sodium chloride so that we don't get aggregation of proteins, and some detergents, some Triton, so that once the lysozyme that we're going to add after we resuspend it in the lysis buffer, once that works on the peptidoglycan layer, then the detergent is there to break open the cells. So all of this part should be very familiar to you from the plasmid prep experiment. Then we're going to, after a certain number of minutes, we're going to add DNase. Now this is in addition to the protocol. And remember, in the aldolase experiment, we cut the DNase step out because the DNA did not seem to be a problem. Well, in this case, the first time that I did this experiment with students, there was so much bacterial DNA that was there that the viscosity of the solution due to the E. coli DNA did not let our glutathione agarose beads pellet to the bottom. So we had to add some DNase to chew up the DNA. So in this case, in the plasmid experiment, remember we were trying to isolate DNA, so we would not have wanted to add DNase. But in this one, we're trying to isolate protein. So we would not want to add proteases, but adding DNase is just fine. So we're going to add DNase. That's going to chew up the E. coli DNA and then reduce the viscosity of the solution. And then we're going to pellet all of that down. We're going to spin it and we're going to pellet down the membrane, and in our supernatant, just like in the plasmid prep experiment, we're going to have proteins and maybe some DNA that's left, maybe some shards of DNA and some RNA. And we're going to add our glutathione agarose beads and allow them some time to bind. And then we're going to pellet the resin slowly. We're going to pellet the resin at only 1,000 RPM in the tabletop centrifuge because the glutathione agarose beads are easily broken and we don't want to crush our beads. So we're going to pellet them slowly, and then we're going to remove off that supernatant. Now we would have saved a sample of our supernatant to run on the gels to say this is what we started with. And after we bind the resin to our protein of interest, to the GST SH2, we'll probably still have some GST SH2 in the supernatant. We're not adding enough pellet. We're not adding enough glutathione agarose really to be able to get all of the GST SH2 out that we have expressed. If you were doing this in the real world to be able to isolate SH2, yeah, you would want to make sure you added enough glutathione agarose. But in this case, the glutathione agarose is pretty darn expensive, and we don't need to use a whole bunch of it just to be able to prove the point. So there'll be some GST SH2 left in solution after we've pelleted the resin, and you're going to just take a sample of that and see all the proteins that are still left in solution, including our GST SH2, after we have bound our GST SH2 to the glutathione agarose beads. Then we're going to be washing with some buffers, a low salt and a high salt buffer, each time, resuspending the beads in the buffer and pelleting slowly in the centrifuge, and then just taking a sample so we can see the proteins that we are eluting off. And if you look at the gel, you will see that they are when you're finished running it, you will see that they are maybe some very high molecular weight proteins. There might be a few. There won't be a whole bunch because our glutathione agarose beads are pretty specifically binding the GST SH2, but you might see a couple bands in there. So then we're going to wash the beads with elution buffer that contains glutathione. And then when we pellet those beads again, now we want to save the supernatant. So previously we've been getting rid of the supernatant after we bound the GST SH2 to it. Like when we were washing, we were getting rid of the supernatant because that was just stuff we didn't want. But this time we need to save our supernatant because now it contains our GST SH2, which have come off of the beads. So you will have saved samples as you went along. you're going to have your supernatant, you're going to have your flow through, which is what proteins did not bind to the glutathione agarose beads. You're going to have your first wash, you're going to have your second wash, and you're going to have your elution. And you're going to be running these on an SDS gel that you will make, just like you did in the agarose experiment. And you will see a band there. It should be about 39 kD total size, because our GST is 27 kD and our SH2 is 12 kD. So then you can judge the results.